Yes, that's a good point. Open this, get a little more light. If you want me to grade your essay in advance, turn that in. It's got to be before Thursday, though, because Friday's the test. If you turn it in Thursday, I probably won't have time to grade it and give it back to you before you go home. I might if you want to wait around after school. But if you want to write it in advance and have me grade it, that'd be a good thing to do tonight. Okay? If write the whole essay, I'll grade it, I'll tell you what's wrong, what else you need, and give it back to you, and you can use that to study for the test. Anyone can do that. Yeah.
You said before Thursday, turn it in?
Yeah. Well, you could. Yeah, if you turn it in Thursday, and the test is Friday. I don't know how I'm going to grade it and get it back to you before you leave. Unless maybe you turn it in the morning, I'll have a couple free periods I can do, or maybe office hours I can grade it. In order to do that. Okay?
Sorry.
Okay, so we're on page 170. We're going to talk about proteins and nucleic acids today. So I'm going to move all this to the side, and you'll keep that in your brain. What is a protein? Well, proteins are big molecules, and they're made up of small units.
Amino acids.
Called amino acids. And amino acids I'm representing with these little circles. So, this molecule is made up of these little things connected together in a line, which I've represented by this thing here. Now, you're probably looking at this going, what in the world is he talking about? Well, let me show you. Let's start small. Let's start with one of these circles, an amino acid. And an amino acid...
Are those the moisturizers?
Well, there are proteins and moisturizers, and proteins are made
I
up
don't know,
of
it just
amino
sounds like a
acids.
bunch of bottles, like with all these amino acids.
Yeah, that's parts of a protein. So that means there's proteins in there. So they're just using fancy language to try and impress you. But here's an amino acid. This is an amino acid. It's got basically 4 parts to it. On one side is what we call an amino group. NH2 or H2N. You could write it NH2 or H2N. It's the same thing. That's an amino group. Anything with a nitrogen and 2 hydrogens attached, we call that an amino group. Okay? So those atoms connected to a central carbon atom. Remember, carbon has 4 bonds? Remember that? And then on the other side of that carbon is what we call a carboxyl group, also called an acid group. Okay? If you ever see this group on a molecule, COOH, that's an acid. And the O is double bonded to the carbon.
Why does that carbon have another bond?
Which one? This one?
Yes, sir.
1, 2, 3, 4.
Oh,
That's a double bond.
I got you.
Okay? Good thinking though. So we have an amino group and an acid group. An amino acid. Do you see? Every amino acid has those 2 groups. There's also a hydrogen atom stuck to the carbon. And then there's your variable group. Your R group is what we call it. The R group is variable, meaning it can be one of 20 different things. There's 20 different R groups that there could possibly be. So that means there's 20 different types of amino acids. And every one of them has a name. Would you like to see the 20 different types of amino acids?
Pretty interesting.
Is it solid?
There they are.
Do you know all of those?
I did. I had to learn these in college. You may have heard of some of them before. This is called valine. This is called phenylalanine. You've probably heard of tryptophan before. You ever heard of tryptophan? That's tryptophan.
That sounds too fancy.
It's hard to see from where you're sitting, but the difference between all of these, they all have the amino group, the acid group, but the shaded blue region is different in every one. That's the R group. The R group is different. And so that's why I have these circles. Each circle is an amino acid. I made this circle pink because that stands for one type of amino acid. This circle's orange because that stands for a different amino acid that has a different R group. Do you see?
Yes, I got it.
Understand.
And what happens is organisms, cells, will hook one amino acid, will bond it to another amino acid. And here we have two amino acids bonded together. What happens is, here's one amino acid right here, here's another amino acid right here, and they form a bond right in between the two. That bond there, it's actually called a peptide bond. It's a very strong bond between this carbon and this nitrogen. So imagine you had two of these. This carbon would bond to this nitrogen. We'd remove those two hydrogens, we'd remove that oxygen, and bond one carbon with one nitrogen of two different amino acids. And now we have two amino acids connected together. So that thing on the right over there, they're calling it a dipeptide, is this. Watch this. That thing on the right is this. Click.
Okay.
Two amino acids bonded together.
That helped a lot.
It does. And guess what? You can add another one on. Click.
Made it big.
And another one. Click. And another one. Click. And another one. Click. And so we're building a protein here. A protein actually consists of probably 400 or 500 of these. Click together in a long chain, kind of like this. This thing has fallen apart over the years. I used to have these all the way through, but they've broken off. I need to glue some more back on.
So what do you call a bunch of dipeptides when they're together?
A protein.
Oh, it's not like a...
No.
Just a bunch of them linked together will eventually form a protein. A polypeptide.
there we go.
Who said that?
Get it, Graham.
Graham said it?
Listen.
Do it, Graham.
You got the props to go from me.
Let's see video footage.
A peptide bond forms when the acid group of one amino acid joins to the amino group of another amino
Can't
acid.
see anything.
In this example, the acid group of glycine joins with the amino group of alanine. The result is glycoalanine. The acid group of glycine loses an atom of hydrogen and an atom of oxygen. The amino group of alanine loses an atom of hydrogen. These atoms combine to form a water molecule. More peptide bonds can form in the same way, creating a chain of amino acids called a polypeptide.
Ah. So here's what they're showing. That right there is one of my circles. And that right there is another one of my
That whole
circles.
group or just one little circle?
All
Like
of
the whole
That whole thing right there is this.
Okay.
That's an amino acid. The amino group, the acid group, and the R group, they're in the shaded R group. And so we call this glycine. And here's a different one. Why is this one different from that one?
That's a different R.
Different R group. So we call this one alanine. So glycine and alanine combine, watch, click, see, and form that. Glycoalanine. You don't have to remember the specific names of the amino acids. But you can imagine if we get enough of them together, that's what they're showing here, that's a polypeptide chain.
Wait, so it's like amino acid to polypeptide chain to protein,
Yes, a polypeptide chain is this long chain here.
which is a
Which is
protein.
awesome.
Well, what this long chain is going to do after it's put together, and we study how it's put together in a later chapter, it's going to fold up going to fold up and fold into a new arrangement,
Which then makes a protein.
which we're now becoming a protein. Here is the folded arrangements that are possible. One type of folded arrangement is called an alpha helix.
Well, that's like DNA, right?
It looks like DNA, but it's not. DNA is actually a nucleic acid that we're going to study in a second. So this folded arrangement, see how it twirls around? That's called an alpha helix. Or you can have it going back and forth like this folded arrangement. We call that a pleated sheet. So you can form your helix or your pleated sheet by taking the long string of amino acids and folding it a certain way. Now we're starting to make a protein. Now remember, this can be 400, 500, even 1,000 amino acids long. So this could be a real long strand. And after this long strand folds up into helixes and sheets, it will continue to fold. This is from the AP book. Your book doesn't really show it good. I'm kind of upset about your book for that. But anyway, check this out. Here's the long strand of amino acids. And here it will twist into an alpha helix. And you can see the little helixes. Do you see the helixes?
I see the helix.
See the helix there? There's another helix there. And then some of the chain will fold into sheets like this. Is folded. See how we call that a sheet when it just goes back and forth and back and forth. There's a sheet right there. See that sheet?
Oh, okay.
And so the whole strand will wind into helixes and sheets and form this glob structure. It looks kind of like this. We call that a protein. So a protein is actually a long chain of amino acids that gets all folded up. That's what a protein is. Question? Anthony, no? Good?
Yeah.
So would it be incorrect to say that a polypeptide is a protein?
A polypeptide is what we call the primary structure of
It's
a
not
protein.
all the way a protein.
It's not a protein until it folds up. So this is a polypeptide, but not yet a protein. Now it's a protein.
So they have to be all like bundled up to be a protein.
It's got to be bundled up to be a protein.
Oh, wow. So if we get like a question on a quiz and it has like that one, it says which one is actually a protein, it'll be that one in the corner.
That one.
Alright.
That's right. You wouldn't really call it a protein until you see that, which we call the quaternary structure. That's AP terms. You don't have to know quaternary, tertiary, secondary, primary. Those are AP bio stuff. But some of y'all will probably take AP bio in a couple of years. Hey, there's a protein right there.
Wow.
Isn't that cool?
That's pretty cool.
Like a person.
This protein has one of my favorite protein names. This guy's going to say it for you.
Are you gonna laugh so you can go with it?
Oh, it's a great name.
Bovine pancreatic trypsin inhibitor is used to illustrate the kinetic nature of biological
That
molecules.
word?
Changes are at the picosecond level.
See how it's vibrating? Remember I said all molecules
Water
vibrate?
molecules.
That is bovine pancreatic trypsin
Why are those
inhibitor.
two like?
That's what I'm going to name my first child. Bovine pancreatic trypsin inhibitor Willis.
Watch
You'll be
it. Watch
kid.
Can we watch it one more time?
Wait, does that mean it's like hot if it vibrates?
No.
Oh.
Yeah, it's warmer than absolute zero.
Good.
Bovine pancreatic trypsin inhibitor is used to illustrate the kinetic nature of biological molecules. Changes are at the picosecond level.
Like tomato juice.
That was who
Note
said
the
that?
behavior
Meredith?
of the yellow water molecules.
Why did those two orange?
If
ones
it's
go?
vibrating, it's not at absolute zero. It's warmer than absolute zero. It's not necessarily hot. It might be hot. I don't know how fast it's vibrating. If it's vibrating real fast, it will be hot. Yeah.
Wait. Okay, they're in that. There are these two orange ones.
Those are water molecules.
Okay. and
They were just showing the size of it compared to some water
Okay, 'cause
molecules.
they just float away.
Yeah, they're just floating around.
I didn't see it.
One more time.
What are the water molecules?
A bovine pancreatic trypsin inhibitor is used to illustrate the kinetic nature of biological molecules. Changes are at the picosecond level. Note the behavior of the yellow water molecules.
Orange.
Yellow, get over.
Yellow, orange.
Pink. Tomato, bushy, stone, structure.
Okay. So those are that's proteins. You ready? The last part are nucleic acids. Oh, and by the way, proteins build structures in cells, and we're going to learn about many types of proteins when we study cells next chapter. But they build things. Your hair is almost completely protein.
Seriously?
Or your nails.
Your nails are almost completely protein.
Proteins build structure in what?
Build structures in cells.
Tomatoes.
What does that mean?
What?
It's kind of mutated.
Bones have some protein, but they're mostly calcium
Teeth.
and phosphate.
That's
My muscles.
Muscles are mostly protein.
That's it.
If you're eating a steak, you're eating the muscles of a cow,
Steak.
which is mostly protein.
What about steak?
Next time, instead of saying pass the steak, next time you're eating, say pass the cow muscles. I'm really hungry for cow muscles.
my dad would pass.
Maybe there's some bovine pancreatic trypsin inhibitor in those cow muscles.
What about chicken
My dad would
fingers?
slap me in the face.
Those are good too. That's chicken muscles.
You're
Okay,
Oh no. Yeah.
I want to give everyone a nucleotide. Well, you're not going to keep it, but I want you to hold it for a second.
What is it?
It's a nucleotide.
Can we use?
Just hold it. Don't lose it. I'll need it back. I'll give that a minute.
Ooh, I got dough.
Your reaction, you got
ribose.
Pretty sure we got all the same one.
Pretty sure if you don't shut it down, I'm gonna shove this paper down his throat.
Okay, listen up. What you're holding now, listen. What you're holding now is called a nucleotide. You need it. This is another word you got to remember. There's a lot of big words this chapter. This is a nucleotide. Go ahead and memorize it. Nucleotide. That's what you're holding. On three, let's say it. 1, 2, 3. Nucleotide. Now, Bennett, up here. My turn.
Sorry.
The nucleotide consists of 3 parts. The parts are a sugar, which is, in this case, is called deoxyribose, but you don't have to remember that.
Yes.
A phosphate, see it says phosphoric acid, that's also known as a phosphate. A sugar, a phosphate, and the thing that's sticking out to the side is a nitrogen base.
I told you all about this.
And here's what the molecule, what you're holding, looks like if you could see all the atoms that make up that molecule. Here's the sugar. It's a 5-carbon ring. Do you recognize that as a sugar?
Monosaccharide.
As a monosaccharide, it's one of these. It's one of these. So there's our sugar. So this molecule actually has a sugar in it. Also stuck to the sugar is a phosphate group. This is what a phosphate group looks like. It's a phosphorus atom with 4 oxygens connected to it. One of them in a double bond.
So then you're in trouble.
So we have a sugar, a phosphate, and in your molecule, in your nucleotide, that's that one and that one, the sugar and the phosphate. Sugar, a phosphate, and what else do we have? This thing sticking off to the side is called a nitrogen-containing base.
I told you.
I call it a nitrogen base, but you can say nitrogen-containing base. Now, if you look at it, let's just look up here. Everybody look. If you look up here at the nitrogen base, it looks like a ring. It looks like one of these, doesn't it?
It's not.
But You can always tell a nitrogen base because it's got a bunch of nitrogen in it. And sugars don't. Sugars don't have any nitrogen in them. So if you ever see a ring structure, like look at this one and that one, and it doesn't have any nitrogen, it's a sugar, a monosaccharide. But if it does have nitrogen in it, it's a nitrogen-containing base,
Nucleotide.
which helps make up a nucleotide.
They don't all-
A nucleotide is a sugar, a phosphate, and a nitrogen-containing base. All three of them together.
3, 1.
Yes, all three of those together make a nucleotide. Now, sometimes the nitrogen-containing bases have two rings. Listen up here, y'all gotta listen. Graham up here.
Okay, disaccharide.
Sometimes it looks like a disaccharide, but if it has a bunch of nitrogens in it, you know it's a nitrogen-containing base. Okay? Now, what do these things do? Well,
Like gives you nucleic acid.
they hook together. Bennett, question?
What if they only have, like, in the nitrogen containing base, what if they only have one nitrogen, not like a bunch?
Sure, there's
Just
no
no-
nitrogen.
They'll always have a bunch.
Okay.
They'll always have a bunch. Good
Good
question.
question, though.
I watch it.
Great.
Now check this out. I've got some of these nucleotides on the board up here. Look closely. What they do is they hook together. Watch this. Click, click, click. And then on the other side, if you take it and you turn one upside down, watch this. Click.
Oh, it fits.
It fits.
That's crazy.
Oh man, I don't have the. Watch this.
That's crazy.
Click. I'm going to replace these two because I don't have the right ones up here. Watch this. Click.
Want to see it?
Like
And
a
wait.
ladder of happiness.
One more.
Not at all.
One more. Watch this.
Or protein.
Click. And what we formed is something that looks like a ladder.
Whoa, that's like a
This is the beginnings of the DNA ladder, which I thought I had in here, but I don't have a picture of it, which makes me very upset. Which means I have to go pull out the big guns
We're making
and
a
show you.
The interweb.
pro look.
No, never mind.
Making a ladder.
Oh, you're making a ladder? Okay, wait. Let me show you what it looks like.
There's your hook.
Computers can do that?
Oh, we're so good.
Oh, behave.
Austin Powers.
Powers.
Chevy Shagan or Shagan.
Austin Danger Powers. Danger's my middle name.
Gary.
Ah! If you take this ladder,
Oh, I see it.
listen, and you make it real long, and then you twist it,
DNA.
that's a DNA ladder. Do you see the ladder there?
No, I can't see ladder.
There's the ladder with the two nitrogen bases hooking together. DNA is a nucleic acid. Just imagine if this thing were real long, you can make it longer by continuing to hook these things together, and then twist it. It's a nucleic acid is an information carrying molecule. Now you may be wondering, Mr. Willis, how does this molecule carry information?
Carry information.
Good question, Drew.
Thank you.
Let me show you. Listen, this is one of the secrets of life. It wasn't discovered till the 1950s. I want you to look at your nucleotides. The nitrogen bases have names. There are 4 names that could be on your nitrogen bases. Adenine, guanine, cytosine, or thymine. Isn't your nitrogen base called one of those things?
Yes, it is.
Now here's how information is carried in the molecule. Check this out. This molecule here is, you can't read it, but that's cytosine, so I'm going to put a C. This molecule here is adenine, so I'm going to put a T, an A. And if I read this molecule going down, and I read the first letter, that's a C, that's a G, that's an A, that's a T.
Cat, cat.
It spells catgat.
Yes.
And if you kept reading it, imagine it's really long, like this molecule here, it would spell cat, and doesn't mean anything to you, but that's the language of the cell. It's like a different language. It's just like somebody speaking French. If you don't know French, you don't know what they're saying. Well, in this cat, that might spell out cat might spell out brown hair. It might spell out tall person. You look the way you do based on what is spelled out in your DNA. If someone was able to rearrange the code, it could change how you turned out. They would, of course, have to do it before you grew up. They would have to do it in the sperm and eggs of your parents. You wouldn't want probably anyone rearranging your sperms and eggs. Might be dangerous. Yes.
Listen to the scientist. You know, it takes someone 96 years just to read your DNA strand. Like,
Oh yeah, it's 6 billion letters long. 6 billion letters. It's like 100 volumes of an encyclopedia, all crammed into a single cell, which is microscopic. This is a long molecule. We're going to study that in a later chapter. Yes, Mary?
Okay, can you like design your child?
Not now, but maybe one day.
Good question.
If we could figure out how to change the letters, maybe one day you could
So
design.
do you know how to do that?
We don't know how to do that yet.
It's on red.
It's on red. Okay, we're almost done.
Okay.
So like if you have like certain like your double joints and stuff, is your DNA gonna be longer? Like if you have certain things that are like,
No, but it's going to be different.
Okay.
It's going to be different. Not really longer, but different.
Cat, cat, cat.
Okay, so
What?
what we're going to do now, listen, what we're going to do now is I'm going to give you another molecule sheet, and we're going to try and identify these. Okay? For tonight, you read the rest of 6.4, and there could be a quiz on 6.4 tomorrow.
There could be.
What about our little
Crazy.
thingy?
All the pictures in
Peace
here.
out.
What about these?
Oh, I need those back.
Press it.
This.
I'll get 'em.
I'll come around.